side? Is it, as the slides moved on? If they, yes, no, someone can just at least indicate that, whether they see the next slide.
No.
The chat says yes. Baruch Hashem. Okay, wonderful. So we talk a little bit about genetics. This is a very, very long subject, but we'll speak about it limited. Gregor Mendel discovered in his garden in his monastery in Austria that pre-plants tend to have traits that are missed out from generation. He recorded that it was some sort of inheritance. Today we call it Mendelian inheritance. The most important databasis is called OMIM, which is Online Mendelian Inheritance in Man. So we call it after Mendel, but really Chazal spoke about genetics as well. The Mishnah, or the Gemara in Masechet Nida says, Tana Rabbanan, Shlosha Shutafim Yesh V'Adam. Everybody knows this Gemara. Everyone knows that there's Shlosha Shutafim. Aviv, but what's less known is that what the Gemara actually says. What did each one give? Aviv Mazriya Loven. The husband, the father, gives white stuff that from comes some of the makeup of a human being. The mother gives red stuff, from which we also get certain things that are related to red or black, or basar, sarod, because both gives the ruach and the neshama. So I think that it's very interesting to think that what Chazal is saying is that what makes you father and mother, we could say many things. Makes you father and mother because you raised them, because you did lots of things. What it says is it's very specific that they gave traits that are somewhat genetic in nature. We don't exactly divide it into white and black today, or white and red, but it's interesting genetic nature. That's also interesting because until not very long ago, we didn't think that the women actually contribute anything. There was an idea that this is Hatzkar's picture under the microscope in 1694, that he said everything, and you can see in the little sperm cell, the entire human being. The man gives everything, and the woman contributes nothing. Chazal said very differently, like very, very long time before, millennia before Hatzkar. And the Gemara, and we're going to come back to this because it's important. The Gemara speaks about in Masachet Yevamot. There was a woman that the Mala Rishon Omed, that she had a first child and she died. The Sheniva mate, Shlishi lo timol, Divrei Rabbi Shimon Gamliel, Divrei Rabbi, sorry, Rabbi Shimon Gamliel says Shlishi mol, Divrei lo timol. So the reason she has one child and another child, and all of them after Brit Milah they died. The third child or the fourth child, Machloket, well, how do we have a chazaka? Was that they shouldn't die. What are they really talking about? It's probably speaking about hemophilia. And hemophilia is a case in which we have the inability to make platelets. And there's a factor 8, which of course to make platelets is missing or partly missing. And therefore the person will bleed to death. That's probably what they had. And what becomes fascinating is the Gemara speaks about it as a genetic abnormality. It's something that we'll see in a moment is passed along families. And we know that that hemophilia is an X-linked genetic abnormality. And the Gemara then continues as Maseh ba'arba chayot betzipori. There was 4 girls in Tzipori, 4 sisters. So Meita Rishona mate, Meita Shniya mate, Shlishi mate, V'Revi Balifnei Yom Shem Nigel, V'Mala Al Tamuli. Why the Achayot Mechaskot? Rabbi Gamliel says don't do the Brit Milah because Achayot Mechaskot, because there's a chazaka among sisters, and that was really what we're speaking about. There's an excellent genetic abnormality will be carried by women, and so these 4 sisters had this possibility, and the men will be affected, which is almost exactly what is described in the Gemara. What's fascinating, the Gemara says, doesn't say let's check out this kid, but the cousin, since he's the cousin of a child who had hemophilia, he may have hemophilia. We know how true this is. So Chazal had a little bit of understanding about hemophilia. And the Gemara is similar. Gemara says, Lo yisa adam isha lo mishpachat nifim lo mishpachat mitzorim. A person should marry a woman from family of Nichvim, a family of Mitzorim. It doesn't say don't marry a woman who has Nichvim. Nichvim is epilepsy. Or has Mitzorat. Don't marry a woman that comes from a family who has that. And the Gemara says there are 3 cases of that happening. And the Gemara also says in the Gemara a tall man should marry a tall woman. They have a very, very tall child. A short man should marry a short woman. As they have an etzbei, they have a very short child. Levan lo yisa levana, a very light person should marry a very light woman. They have a very, very light child. And a dark man should marry a dark child. What Chazal understood well ahead of their time was that there are genetic traits that are amplified from generation to generation. If we have a very small gene pool, we're going to have an amplification of those things. Now, being light or dark or short or tall are not necessarily genetic abnormalities, they are genetic traits. But what Chazal worried about, not just don't be sick, don't have nishpachah nechvim, nishpachah matzarim, but try to recognize that there is an advantage. It is advantageous to introduce other genes into the gene pool, and that's very important. So, what are genes? So, genes are basically the map that contain the material, the information, in which we will be able to create a human being. Not we, but the Kadosh Baruch Hu, the body. 23 chromosomes come from the egg. 23 chromosomes comes from the sperm. And when they're put together, we have a full gamut of cells, which is basically what we all are. And we arrange them rather unexcitedly into pairs. They're called 1, 2, 3, 4, 5, 6, based on the amount of information. So 1 has more information than 2, has more information than 3, etc. Until we get to 21, 22, and then we have X and Y, which makes you either male, XY, or XX female, or more complicated genetic abnormalities. Now, each of those genes has an information, is a wealth of information, much more than we get in the book. It's having each gene is many volumes of a book, incredibly well and compacted information. Sometimes they have on a computer disk, or some sort of digital information. It's very compacted into a very small place. But we've been able to map those genes. So on gene 15, for example, you can't see the slides I'm showing at all. No.
We only see the first line.
All right, so we'll go back. Let's go from here. You can see, what are you seeing now? Gene 15?
Yeah, now we see it. Yeah.
And now, what is it? If I go on, we're seeing trisomy 21?
Yeah, now we see. Yeah.
Sorry, you told me before. I was getting excited. I thought I did something well. Now I see I didn't. This is a genetic abnormality, being an inability to share your slides is a genetic abnormality. So gene 15, for example, we only have, we've mapped all the genes. We know on gene 15 is very important for us Jews. We have Bloom syndrome, we have Tay-Sachs, we have Marfan. We know the markers. Now that's very important when we think about that concept that we know genetic markers, we know where they are, because those genetic markers will be the indication that we have abnormalities. Now that's very important when we come to eradicate those genetic abnormalities, which you're going to speak about later on. That's the whole point of what we're going to speak about in the next few minutes. There's other things, trisomies. Trisomy, the most famous is trisomy 21. We call it Down syndrome. Down was a horrible person. He was just an anti-Semite and just a particularly unpleasant person. And so we like to call it trisomy 21 and not Down syndrome, but there's the same thing. And trisomies are basically, you have, instead of a pair, you have a 3. That's not a good thing. You have a trisomy in any of the green pairs. Here's 21. Now 21 is very fascinating because it's the smallest gene pair, almost, and yet it has so much ramifications, just having a little bit more. You have trisomies in all of you have trisomy 1 and 2 and 3. Trisomy 18, some people may have heard of because you can be born with it, but it's not compatible with life. Children die with it. But many of the trisomies, they will sort of develop. You'll have they'll miscarry. We have monosomy, like here. We have XO. There's nothing in the X. That's what we call XO is, there's no Y, there's no other X. That's either Turner syndrome or other syndromes in which we have, we're missing some of the genetic material. Here's a monosomy 22. You see 22 is only 1. It's not a pair. We can have terataploidy. We can have lots of 4 pairs, in which is definitely not compatible with life. So there's not just genetic abnormalities other than just things that are inherited. You can't inherit Down syndrome. They're very simple because Down syndrome men are not fertile. And so they can't pass on to their children because they won't have any children. The other thing to remember, what's very important, is that as we get older, as women get older, not men, as women get older, there's a greater chance of aneuploidy, greater chance of having embryos which are genetically imperfect. And therefore, there's a greater chance of having, of course, miscarriage. So we look at the maternal age on the bottom axis and the amount of aneuploidy we have. There's a clear increase as you get older. And there's obviously a clear increase of miscarriages as you get older for the same reason, because most of those miscarriages will come from aneuploidy just even make it. It's not that they're born with genetic abnormalities. They just won't develop at all. And of course, obviously, you have a corresponding decrease in live births, because the more miscarriages you have, the less live birth you have. So how do we move genes on? So of course, there's recessive genes, things like Tay-Sachs, which is a recessive gene in which the mother and the father both have to be carriers, not to be sick. They have to be carriers in order to pass it on. And they have a 1 in 4 statistical chance of having a child with that genetic abnormality that we said, like Tay-Sachs. There are dominant genetic abnormalities like Huntington's, which is a dominant genetic abnormality that you don't have to have 2 parents with Huntington to get Huntington. And if you can't be a carrier. You get it, you're sick. Now, you might not have all the symptoms, but you'll get it, you're sick. And you have 50% chance of having your children get it because you're going to pass on here's the affected father, he's got Huntington. He has a 50% chance of passing on to any of his children, male or female, it's the same. And there's another genetic abnormality called an X-linked genetic abnormality, which is only carried on the X gene, like hemophilia, we just spoke about, or Fragile X, which is only carried by women. Women, since they have another X chromosome, usually are not sick. So a woman with a Fragile X won't be sick, but if she has sons, they'll be sick. They'll get that genetic abnormality. That's very important to think about. And we're rushing through this very quickly, and you'll listen to the tape again later and look at the slides again. But it's very important when we think about genetic abnormalities, what are we talking about? Because the way we can deal with them is very different if it's recessive or if it's dominant. Because recessive, we can just say, don't get married. If it's dominant, we can't say that because whoever you marry, you're going to have it. X-linked, if you have only daughters, we can solve the problem. If you have sons, you don't solve the problem. So it's very important to understand how it's moved on. And of course, that's how we get to genetic testing. And of course, premarital genetic testing, such as Doi Sharim, which I'm sure you're all familiar with. The Doi Sharim, 40 years ago, changed the map of genetic testing across the world, not only for Yidden, for everybody. And every single genetic counselor in the world will know Doi Sharim, because Doi Sharim really was the first to speak about, let's just check everybody for recessive genetic abnormalities. And it was talking about Tay-Sachs, and we will eradicate all of those genetic abnormalities. And Rav Moshe and other poskim came out strongly against it initially, because they said you'll create mishpachat nichfim. Remember, we just said don't marry mishpachat nichfim. The more we know that there are genetic abnormalities, the more mishpachot nichfim you've created. And so Rav Moshe and others said you shouldn't do it unless you have some sort of a code. And what you get a serial number. Instead of getting an answer yes or no, you get a serial number. Now, we all know how Doi Sharim works, and Doi Sharim, of course, has changed the Jewish community, and has eradicated basically Tay-Sachs and other genetic abnormalities in one generation. Very simply because if you're a carrier, you're not sick. And that was what the poskim had to understand. And now we all understand that being carrier is not being sick. But once you're a carrier, doesn't marry a carrier, and it's a recessive genetic abnormality, you've solved the problem. Of course, that doesn't help if you have a dominant genetic abnormality or you have an X-linked genetic abnormality. We can't solve that problem just by not marrying somebody, because whoever you marry, you're going to have the same chance. And so that's what Doi Sharim is very important, and it has it was the first, but it may not be the best. There's a whole question, do we have open or closed testing? Even if we're talking about only recessive genetic abnormalities, some people, I want to know that information. I want to know because it's important for me to know, because I'm in a community in which knowing is important. There's a question whether Doi Sharim is good enough for Ashkenazim and Sfardim, or good enough for Ashkenazim and Sfardim marrying each other, which obviously has a different gamut of different genetic abnormalities. And it definitely has it's a limited panel. You can do a much wider panel. It definitely serves a purpose. Its service is to prevent problematic marriages. You can go through Doi Sharim and still have a serious genetic abnormality, and you will not have picked it up in Doi Sharim. Why? Because if you've got Huntington, they don't test it. Why? Because they don't care whether you have Huntington or not, because whoever you marry, you'll still have a chance of passing it on. That's a very important point, because many people come to me and say, we're pregnant, we've done prenatal testing, or we've done we're testing in prenatal testing now, and we've seen the baby has some problem. They say we have to go and do amniocentesis, because we want to see the genetic makeup of the child. But we've already done Doi Sharim, so there's no reason we should do that. That is just completely flawed logic. And it's logic that's based on a complete misunderstanding what Doi Sharim does. You can go through Doi Sharim and still have a genetic abnormality. You can have done all the testing and still have a child who's got a genetic abnormality, because there's aneuploidy, because there's things that are not hereditary. So not all genes are hereditary. And that's very important when we think about Doi Sharim, what it's for and what it's not for. And therefore, of course, you can go to a genetic counselor and you can get genetic counseling. You go through JScreen or any of the other companies that'll do large gamuts of genetic abnormalities. We don't always need that. We may actually be have too much information. Mosif machov. Maybe there's too much information we can get by testing every genetic abnormality that we have. And you can look at what is the chances of having a problem? What is it? What is the frequency of such an abnormality? But also, what is the ramifications of such an abnormality? So all of the list of here, Tay-Sachs, Canavan, cystic fibrosis, Fanconi, Niemann-Pick, all of these are very serious diseases that have great ramification, most of them also fatal. Gaucher, you can live for a long time with Gaucher. Rav Steinsaltz just died a year ago or so, as it's all, and Rav Steinsaltz lived a long and very, very productive life with Gaucher. And so, but it has an impact, and you have a chance of passing it on. And so it's important to think about the frequency and, of course, the severity of the disease when we speak about counseling people to test A or B disease. And of course, there are diseases which are very common to the Jewish community, with the Ashkenazim have the greatest number of genetic abnormalities of any ethnic group in the world, not only in the Jewish community. So if we have the genetic abnormality race, Ashkenazim win, hands down. That's not necessarily a good race to win, but we run it out now. And there's a long list of other things you can test, and there are 1,000 things you can test, and we have to think about whether that's something we want or not. There's, of course, other genetic abnormalities like BRCA. BRCA is a genetic abnormality that causes cancer. Basically, how cancer works is that everybody has cancer. Cancer is basically a proliferation of cells, which is unchecked. So our cells, as we're sitting here talking or listening, our cells are replicating themselves. That's what happens the whole time. And sometimes that gets out of check, and those cells go wild and start replicating. And the body notices that, either fixes it or kills off those cells, and that happens the whole time. That's a normal process of having micro growths that basically micro tumors that basically we deal with. BRCA, that ability to identify those problematic cells is less developed, and therefore those people, those women and men, but mostly women, will have a greater chance of getting cancer. So if we look at BRCA 1 and 2, the incidence of breast cancer increases from about 12% to almost 85%. And ovarian cancer from 1.4% to somewhere around 25 to 40%. And it's a familial thing. So it's a chayot mechaskot thing. So if we have BRCA in the family, it's inherited. And in the normal population, it's about 0.5%, and in the Jewish population, 2.4%. So we're 5 times more. I think it's less more. We're even more than 5 times more likely to get it than anybody else. So these are genetic things that are important. So there's another thing we do is prenatal genetic testing. We can test before you got married. We can test before you have your children, before you have babies. So we're talking about eradicating genetic abnormalities. And the thing we can do before we do anything is to just test before you get pregnant. So we can take cells from the placenta. We can take cells from the amniotic fluid, and we can look at them. We can see whether that child has genetic abnormality or not. You know, when an amniocentesis, and then we can amplify that material to actually be able to see the genetic makeup of that as yet unborn child. Because once we have this material from the placenta or from the amniotic fluid, we have the cells of the baby. And so in the 50s already, we started having the ability to look at karyotype, which is about let's say a 10 megabyte piece of information. That was the most we can get. In the 1990s, we had something called the FISH, which got to now 50 kilobytes. We're going to come to a much higher resolution. And the 2000s, we got to microarrays, which are about 2 kilobytes. And today, microarrays and next gen, we're already getting down to very, very small pieces of information. We have a tremendous amount of information we can check. So if I do today an amniocentesis, or I do a CVS, chronic villus sampling of a baby, I can basically get to a very, very exact amount of information. I can do a complete scan of that baby's genetic makeup, and we can see where they have a genetic abnormality. And here we've seen, look, you can see, this is looks at, we take a control and we'll take the abnormal, we can see this is abnormal. Here's a genetic abnormality. There's a translocation. There's a piece of the gene missing. There's a piece of the gene which seems to be in the wrong place. These are information we get, and we get this the whole time. And the question that we have we've got an incredibly high resolution. The question that we have is, we're getting onto exome sequencing, we're getting onto next gen. Should every woman who undergoes a amniocentesis be checked with a chip? Should every woman of any age be advised to undergo genetic testing with a chip? Should we be doing more and more genetic testing? Should we? And we're talking about pregnancy. We're talking about prenatal. We're not talking about premarital, which is the whole issue in of itself, as we just said. And we're not talking about pre-implantation genetic diagnosis. We're talking about a woman who's pregnant. So a woman undergoes an amniocentesis because she's over 35 in Israel, or over 38 in England, or because she's got a good insurance in America, or because she saw something abnormal in the first scan, and now she's got an amniocentesis. The amniocentesis, if you do a regular amniocentesis, will come back okay. But I can check it with a chip, and I can get a tremendous amount of information. Or let's say even more than that, everyone at any age, you should all go and do genetic testing, because we'll find out that we have babies with problems. And what we have, and what we will come as a Rabbanim more and more to face, is something called the VUS. And if we take a chromosomal microwave of a baby, of a fetus, that's your patient, right? We have two possibilities. There's normal, abnormal. Normal means you've got a normal, complete set of chromosomes. And abnormal means you've got trisomy, you have an aneuploidy, we have a problem. But there's actually something that's in between those two. It's not a yes, no answer. It's not a green, red. There's the orange, which is the VUS. The VUS. VUS is DOS. It's a variant of unknown clinical significance. And the variant of unknown clinical significance is, I got an information. Rabbi, I went to do an amniocentesis. They did a chip. And this is what I got. And this is the answer. What should I do with this? Can I terminate the pregnancy? Can I continue? What should we do for later subsequent pregnancies? And the answer is that while there is a tremendous amount of information in our databases, we don't know every single genetic abnormality. We don't know what that means. And I'll give you one example of the VUS challenge. This is a case study that I was personally involved with here in my city. We have a woman that's 42 years old, and she had 6 pregnancies, 1 miscarriage, and 4 healthy children. The youngest is now 4, and she's 42, so this is probably the last pregnancy they're going to have. There was a slight increase in amniotic fluid. The ultrasound seemed to be fine. At 31 weeks, there's a slight IUGR, intrauterine grade 3 retardation. The baby's a little bit small. So they went to do amniocentesis at 32 weeks. Now, 31 weeks is very late in the pregnancy. The result is no more correct, but we have, of course, a chance in amniocentesis of having a miscarriage. And this is the result that they got. So I don't know if you can see that, but you can look at it afterwards. But they have a chromosomal abnormality in a very, very specific place, a very specific marker. And the problem with that is, in all of the gamut of the literature that she went through, the doctors went through at the time, there were 3 cases in the world of somebody that looked similar. And all the 3 cases terminated the pregnancy, because when they saw that, they got cold feet and they terminated. So we have no living children who have this. And the question is, what do we do with that? Do we now know that that's normal, abnormal? And the answer is, no one knows that information. And what happened is, a little bit of the background story to this story was just, she called me on a Thursday night and said, I have this problem, and what do we do? I said, okay, send me the paper. I've actually got a chavura, and I'll go after my chavura. I'll look into it. And the wife of the person whose house my chavura on Thursday night is a doctor. So I said to her, look at this paper. What do you think? So how do you get that paper? That's a patient of mine. That's my patient. I saw that patient today. Okay, what do you do? She says, I have no idea. We have no idea what to do with that. And I went to meet with a couple. And in the end, I said what they said, and they did terminate the pregnancy. But nobody knows whether that baby would have been healthy or not. And that's the VUS challenge. That's a lot of, when we think about genetics, we think it's always black and white. It's always Tay-Sachs or Down syndrome. The reality is, in our generation, we're getting closer and closer to the point in which it will be more and more VUS, more and more questions that we really don't have an answer. No one has an answer. So it's very important to remember that. And there is a lot of databases. There's OMIM and there's Franklin, and there's other databases that we use to try to get that information, but that's really something you need to go to an expert to see. Which is why there's another option, which is pre-implantation genetic diagnosis. That's not prenatal. We've gone from premarital prenatal, i.e. during pregnancy, to the thing that really is related to what we've been speaking about in the last few weeks with IVF and doing fertility treatment, is that, well, if we know there's a genetic abnormality, we could do an in vitro fertilization and we could check the embryos before we put them in. Because remember, every embryo I create in a lab contains all the genetic material that's ever going to be in that baby. So by removing, as you see in this very stylistic picture here, by removing a cell and analyzing the DNA of that cell, I will have a full picture as though I have a person in front of me, as though I have a patient here. And that's very, very important to remember. But that only is helpful if I know there's a genetic abnormality. And let's say we have an example of someone who has a recessive genetic abnormality, gets married, or somebody who has a dominant genetic abnormality, or someone has an excellent genetic abnormality. In all three of those cases, I can't now tell them don't get married. They're already married, or it doesn't matter who they marry. And so what we can suggest is to do PGD. I'll tell you a story that I was with Rabbi Siegelman once. I flew into America at 6 o'clock in the morning. And I touched the ground running, so I'm already after davening, already teaching and working and meetings. And it was like 8 o'clock in the evening. I'm supposed to give a lecture. I think I'd given a lecture, and then I was going to have Marvin, and that was it. It's turning. And a couple said, can we just have a word with you? Sure. So Miss Marv, and I spoke with this couple for 2 hours, and they had just got results from something like Dori Shurim. It wasn't Dori Shurim because they wouldn't have known this, but they something like Dori Shurim, that they were both carriers of Tay-Sachs. So what do? So don't get married, but they want to get married. That option of not getting married wasn't really relevant for them. And so the possibility that we suggested after long discussion and my input that they shouldn't get married was a possibility to do PGD. So let's understand how it works. So we do IVF. Now you're all experts now in IVF, how it works. We all know that. So we create IVF, and then we take out a cell or a number of cells, and this is developing over the time. This is sort of a history of PGD, and we can do either FISH or CPR, or we can PCR. We can now do, of course, next generation. We can basically do an entire check of the embryo and tell you what propensities, disease that embryo would have, and what possibilities we can do with that, and what will happen. Basically, we can get an entire genome as though we have an individual in front of us. And the question, why should we do it? So the answer is, it's exact. It's a very, very exact science. We can tell you that, what the genetic makeup is, what it have. Of course, you have the VUS. We have the uncertain significance. That's true. But it's only an embryo. It's only a fetus. It's not even a fetus. It's not even an embryo. It is a fertilized egg, and that distinction is very important. It's not an embryo. It doesn't have arms and legs. It's a ball of cells, so we can throw it away. It's no question of it's Maya Ba'alma. We'll come to that in a moment. It's a developing science, so that as we've seen even in the previous slide, it's developed from our ability to analyze 10 years ago or 20 years ago when I first started working in talking with PGD, to what we can able to do today. We're doing it much higher resolution, much higher importance. And it's got multiple uses. We can, of course, look at lots and lots of genetic abnormalities in one go. Why should we not use it? Why shouldn't everybody do PGD, or everybody over a certain age do PGD? And the answer is it requires medical treatment. So this is a medical treatment, as you just learned in the past couple of weeks, that doing IVF is not a fun activity, and it's not a particularly easy activity, and it's also not a particularly cheap activity, and therefore it's going to severely restrict the number of children. We're not going to have numerous children through PGD. There are side effects of doing PGD, side effects of the mother taking the hormonal treatments, doing IVF. There's also side effects for the baby. We're removing a cell, we're removing a number of cells that has an impact on the fertilized egg, and it's not 100% successful. There can be mistakes. There's something called mosaic, which means that some of the cells are express one type of gene, and some of the cells except other type of gene, and therefore we may be doing, especially only taking single cell, which we're really not doing anymore, but single cell PGD, we could get a mistake. We can get a mistaken read, or we can get something that's unclear of the significance. And it's, of course, lots of ethical and halachic questions of doing PGD. So the halachic questions really have are, okay, what's the status of that embryo, that fertilized egg? And are we allowed to undergo an elective treatment? And what are the boundaries of what's permitted? Like, what is the gvulot of what HaKadosh Baruch Hu wants of us? So Rav Elyoshev said a very, very important distinction. Rav Elyoshev Zatzal said to us that the distinction should be the following. If it's used for medical reasons, it's mutar. And if it's used for cosmetic reasons, then it's asur. So when we get into the question of what is the gvulot, what are the limits, question is simple. It's a very simple. Medically, if it's in a medical reason, let's say a couple of both Tay-Sachs, so then it's a medical reason, as opposed to I want to have a child who's we want to have a specific type of gender, a specific type of child. That would be cosmetic. So that was Rabbi Yoshev's distinction. We're going to come back to that distinction moment. The question of what is the status of an embryo? That's very possible. We have a Gemara. The Gemara says, Gemara Masechah Yevamos and other places says Arba'im until day 40. My balmo, it's just considered water. It's not really water. It's cytoplasm, but it's got the din of water. It's the din of something that doesn't have life, and therefore we don't think of fertilized egg as a life, unlike Catholicism, which does, and therefore in Italy it's very problematic to do PGD because you are not able to discard in Italy frozen eggs, fertilized eggs, whereas in Israel and in the Torah and in Halacha we are. So it's basically not considered it. So Rav Elyashiv's very interesting distinction could be questioned when we actually put it down to practice. Let's take example, PGD for a carrier. I am a carrier of a genetic abnormality. Let's say we'll call it Tay-Sachs, because it's the most common. 1 in 25 Jewish Ashkenazi men are carriers of Tay-Sachs. So it was very difficult for me to find a shidduch because the first 3 girls that I went out with were also carriers. 1 in 25, they were also carriers of Tay-Sachs. And I got married very late. And I don't want my children to go through that. So I want to do PGD, not to have a child who's healthy. I want to have a child who's not a carrier. It doesn't have any genetic abnormality. Is that what that? So on the one hand, it's not ill. It's basically cosmetic. But there are ramifications for the future of A, your social standing, and B, there are certain genetic abnormalities in which being a carrier does have an impact, does have an importance. And therefore, can we say that that's a medical thing? Or is that only cosmetic? Cosmetic is only when we're talking about choosing particular traits, or is choosing not to be a carrier also a trait? It's not a medical need. And so this is a big, heavily debated question in the genetics world as to whether we should be doing PGD for carriers. And most units will not do PGD for a carrier. They'll say, no, you don't have to do it. Even if a couple anyway are going under IVF. Now, if a couple anyway are undergoing PGD for something else, they might say, so we'll also check all the embryos and only put back ones that are really healthy. But that's generally not the case. Of course, the most difficult question and the most not uncommonly asked is gender selection. The Gemara gives us a formula for making sure we have males or females. So the Gemara itself wanted us to make sure we had male or female children and to be able to influence their gender. The Gemara speaks about bo'el v'mashaneh, bo'el v'shalesh, speaks about eating certain things, about timing of intercourse, and therefore there's not a problem ethically with choosing I want to have one child, another child. We're able to do PGD very, very simply. It's a very easy thing to check whether the child has an X chromosome and a Y chromosome or only X chromosomes. Very, very easy to check. So medically we're able to do that. Should we be doing it? So gender selection, of course, is a very, very difficult question because it sounds so cosmetic. What do you want to do? I want to have a boy, I want to have a girl. I want to have, and we get questions from, a couple we have one boy, we can have one more child, and we want to be a girl. And so we want to do gender selection. So that's, Lo Yeshvim Tov doesn't sound too well because of what Rabbi Yosef just said. But there are, of course, cases in which gender selection is not so pashut. We'll take 2 examples. One example, I was in a community somewhere in North America, and there was a couple there who had 3 children of the same gender, and the wife had become incredibly depressed. She was incredibly depressed, and as each child was born of the same gender, she became very, very, very upset, and really she was taking medication, and later she was hospitalized. And it seemed that the best thing that they could do is would be able to have a child of the other gender. And so is that permitted? That but then becomes cosmetic or becomes medical. On the one hand, we could say to them, that's completely cosmetic. The fact, go to see a psychiatrist, go to take care of your problem of getting depression because you have the gender that you didn't want. But on the other hand, she was suffering from depression. And so it would seem to be that having the child of the other gender would have been the best therapy for her. So this was an ongoing discussion. In the end, they did get permission to do gender selection, and they then had 3 children of one gender and 1 child of the other gender, and they were ecstatic. And I actually went to visit them one Friday morning, and it was just beautiful to see how her acceptance of all 4 of her children was incredibly altered by the balancing of her family. And it's hard then to say that that's just cosmetic. That's almost becomes now somewhat a medical treatment. We could, of course, argue with that. And I've brought this case many, many places. And generally in the crowd, you always get people who that doesn't sit with them well, that example. Just as a piece of information, I met them on a Friday, and that Shabbat I was in a community not very far from where they lived, and they told the story because it touched me. Of course, I changed a lot of details. If I changed a lot of details here, you wouldn't recognize who it was. And her husband, I didn't know he was there. He came up to me after the talk and said, "A great talk, Rabbi. You know, my wife has a very similar story to the one you just described." So I realized I changed enough that it was good enough. So that's another case of gender selection, which we do have those cases. I give you one other case, which is a very, very famous case in Israel that I was personally involved with, which was a couple, Mr. and Mrs. Cohen, who live in Yerushalayim, and he had a genetic abnormality, which means he had no sperm. He was azoospermic, and they got a heter to do sperm donor. And I don't want to get into the sperm donor debate because that we're going to speak about in a couple of weeks when we speak about the sperm donor itself. And the heter, the Rav Moshe, like them, I'm sure many of you know that Rav Moshe's heter was only with non-Jewish sperm. And so the child, of course, that was born would not be a Kohen if it was a boy. If it was a girl, it wouldn't be a Kohen, but that's not enough Kamina, which would mean that when this man came to have, in 13 years, when the child became bar mitzvah, he wouldn't get an Aliyah Kohen, and he wouldn't go to the Birkat Kohanim, and he wouldn't eat Truma, and all sorts of other ramifications not being a Kohen. And everybody say, how come Mr. Cohen's son is not a Kohen? And then it would come out that he was a sperm donor. And he was incredibly embarrassed from this. And he wanted to get a heter to do gender selection to have a girl. And cut a long story short, they got a heter and they did it. And the doctor was so impressed. It was the first case of gender selection in Israel. They were so impressed with what he did. He went to the press and said, "Look what I did. Mr. and Mrs. Cohen from Jerusalem had a sperm donor. They didn't have a girl. They wouldn't have a boy because it wouldn't be a Cohen, so they'd have a girl." And it's on the front page of the news. And the guy, of course, is livid. Now my whole, it's going to come out, that my whole thing, that I wanted to keep it aside. Everybody's going to know Mr. and Mrs. Cohen. I said, you're right. So I pulled down the phone book. It used to be phone books, you have big things. And I was phone book of Yerushalayim, and I said, how many, there were 18 pages of Cohen. 18 pages of Mr. and Mrs. Cohen. I said, okay, it's one of these. I don't know which one it is, but it's one of you, right? It's one of these. So he didn't speak to me for a year. He was really, really annoyed. And then I saw him later, about a year, and he approached me and said, when can we do it again? They have 2 girls. So is that medical? Is that cosmetic? I don't know. It's very hard, again, to come to give a decision as to whether anything is purely medical or purely cosmetic. And I think gender selection is a very good example, because generally the public will tell you, yeah, of course it's cosmetic, and we just say, give 2 examples, which maybe it isn't. Very difficult answer. Huntington's disease. So let's give the case, a very, very case. It's not a halachic thing. It's an ethical thing that the geneticists discuss, which is that a man's father. So the question about sperm spinning and all of these other questions, all these other amples, I think that the poskim didn't like them at all, because it's also a question of Otzazar Vatala. You have to realize that they're also very, very ineffective. Basically, their chances of success are very, very small, and therefore, we're not really suggesting them. So really, what was really debated in earnest was the PGD, and the PGD has other ramifications as well, because again, it's much more invasive than doing an IUI, but IUI would be also similarly halachically problematic. A man has Huntington's disease. He has a son. Huntington's disease is a brain wasting disease that you get into by you're born healthy, and then by the age of 50. Many end up in a wheelchair and very, very severely disabled. So a man has a son, he's got, his father's got Huntington, and the son doesn't know whether he has it or not. He doesn't want to be tested. So he goes to the doctor and says, do PGD to make sure my children don't have Huntington. It's too late for me. But don't tell me whether I've got it or not. I don't want to know. And the doctor starts doing a PGD and sees that the child, that none of the embryos have Huntington, because in fact, the son of the father, the father of the embryos doesn't have Huntington at all. So he tells him, you don't have Huntington's. So the guy's, of course, fascinated. He's ecstatic, and he doesn't have to do it anymore. But the next guy comes in, he has exactly the same story, and he doesn't tell him. But what does the guy now know? He now knows that I must have it, otherwise you would have told me like you told the guy before. So the question, can he continue doing PGD for the first guy to save the other guy, to save the other patients? The question is, who's my patient? That's a very big ethical question, which I don't really think we have an answer. But it's a question that's highly debated among the geneticists. And think about a thing like BRCA. But we can do PGD to remove the BRCA mutation. Do we have to do it? Are we allowed to do it? BRCA is usually something that's what we call late onset. So it's not something that happens now. It's something that happens in 40 years' time. And in 40 years' time, the child born today, we will be doing, I don't know. I have no idea how we'll treat cancer in 40 years' time. I know how we treated cancer 40 years ago, and I know what we've got to today. So I can imagine in 40 years' time, we'll treat cancer much better. So should we be doing PGD now to get a child in 40 years' time won't have cancer, which one time cancer may be like having, a flu in which you'll just take a shot and you won't get it. We don't know. So that's a big question. We don't know. And that's a huge debate. Should we be doing it or not? Which you will get I have in my shul, one man and one woman, they're not married to each other, who are BCA characters that I know about, that are of reproductive age. This is, something that's common. We could do another thing, which is not only PGD for diagnosing a specific illness, but we could do PGS. We can screen all embryos. Now, if we screen all embryos in IVF, IVF becomes the norm to do all screening everybody. And you're in America, many of the labs in your vicinity will be offering PGS because it costs, it costs you $5,000 an embryo, and you can do PGS, and we'll screen the entire genome. Very smart. But we will also discover a lot of things which are vusin, which you don't know about. Also, the other thing is that embryos generally have the ability to correct some of those genetic abnormalities. And so we may be erratic, we may be throwing away and discarding embryos which in fact are perfectly healthy. Just we can't see that they're healthy because they look sick at the moment, but actually they're healthy. And so we have to be careful to suggest do PGS. PGS is very good for women who are older. It's very good for women who've had previous miscarriages, but it's not, generally should not become the norm. And what we have to be careful about is not getting the perfect child. We're looking for the perfect child because there isn't a perfect child. I said to somebody the other day, you have to recognize that if you do PGD, you're not going to be ensured that your child's going to be healthy. You're going to be assured that there's a good chance your child doesn't have the genetic abnormality you look for. But we can't ensure your child's going to be healthy because there is no perfect child. There's actually a very nice book that I recommend if you're interested in, something to read. Ben Minchel Amariv on genetics is a book called The Survival of the Sickest by a guy called Sharon Ma'alim. He has a very interesting proposition. His grandfather died of hemochromatosis, which is having too much iron, and you get iron deposits all over the body. And his suggestion was, why do we have this? Why is this still around? Why did HaKadosh Baruch Hu or evolution allow us to have the genetic abnormalities? Why wouldn't it just eradicate it the generations ago? And he says maybe there's an interesting idea that maybe in the Black Plague, we saw that some people died and some people didn't die. Why did people die who seem to be healthy, and people that didn't die seem to be unhealthy? Look at Corona. Look at all those other things. My wife has an uncle who was on death's door from Corona and got up and is now fine. I was dancing at his grandson's wedding with him a couple of months ago, and the doctor said, "We've never seen someone of your age get better." Why is that? So he said, maybe having too much iron, in fact, now is beneficial. Maybe having too much iron is actually an antidote to getting plague, and therefore, as the Rambam says, the Vayadua B'olam says the Rambam, mate Bali. Nemenet, a woman says, my husband died. Nemenet, Shadavar Pashut, Befi Kol Adam. Everybody knows this. You have to be a Rambam. You have to be the doctor. Everybody knows. Kol Adam, Shebishnat Dever, Zechai Vezemet. Some people live and some people die. They're muted bedever na'arim chazakim v'yinatzlu zkenim acholim. Some of the young people, we don't know why. There's no seems to be rhyme or reason. Says Mu'alim, maybe it's because you have too much iron. So maybe there are things that are antidote. Bloom syndrome, which we mentioned before, is one of those things that was checked by Dor Sharim. In fact, I was very instrumental in getting it included in Dor Sharim because of the case that we had. A guy who had Bloom syndrome. Bloom syndrome is on the 15th chromosome, like all the other some of the other diseases that we mentioned today. And Bloom syndrome causes a lot of cancers. And it also causes infertility for men. So a guy came to me many, many years ago, and he said, I have Bloom syndrome. I didn't know too much about it. And in fact, there's a register. Then there was a registry of all the people in the world that have Bloom syndrome. And it was in Cornell University. A guy called James German had the. And I went to see Professor German, and he told me, he said, it's very fascinating. First of all, a lot of them are from Jews because they don't terminate pregnancy. And he said the amazing thing is that these men get a lot of cancers. They don't get prostate cancer. And prostate cancer is the most common male cancer. Why they're not getting it, we don't know. What is the antidote in Bloom syndrome to prostate cancer? I.e., we have to understand that the Kodesh Baruch Hu has created this idea. The genetic abnormalities, some of them, in fact, are antidotes to other things. So sickle cell anemia is an antidote to malaria. And other things are, if you have this, then you won't get that. And that sometimes is beneficial. There is no bad gene. There is a gene that we have an expression that we think is negative. But in fact, there may be, and there probably is a positive, that otherwise it wouldn't have survived through a Kodesh Baruch Hu's world or through evolution. So that's what we can do at the moment. What we can do PGD. And PGD is a very fascinating procedure. And it raises very good questions. Should we be doing it or not? So we think of Rabbonim. What have we learned so far? So we learned so far is that we've learned a little bit about genetic testing and who should we be doing genetic testing for, and what do we do with the results? The second thing we learned, we spoke a little bit about Chazal and genetics. The second thing we did is that we spoke about testing during pregnancy and what are the ramifications of that. And we spoke about the VUS challenge of getting things that we don't always know exactly what that means. What do we do with that? And the third thing we spoke about was PGD and what are the limits of PGD. And when Rav Elyashiv said medical, yes, cosmetic, no, which is a beautiful and incredibly permissive approach as opposed to other poskim and other religions, of course. That's a very beautiful division, but it's not always so easy to make that distinction as to whether we see this as medical and this is clearly cosmetic. And then to think about that we have to speak about this, that we're not going to get the perfect child because there are people who want to do PGD for things in which I don't think it's warranted to do PGD, to have late onset or to have things which are, I want to get the perfect child, or to do PGS for that matter, to say we'll do the screening and not just look for a particular disease, but we'll screen for every single genetic abnormality. That's maybe too much. And that's where we're at pretty much today in a clinical basis. And where we're at in the lab is somewhat completely different. What I have lab is something called CRISPR. And if anybody follows the Nobel Prize winners in biology, the Nobel Prize winner of last year was Jenny Dover. She won for her work in CRISPR. What CRISPR does, CRISPR-Cas9, is that CRISPR stands for clustered regularly interspaced short palindromic repeat, in which we are looking for the genetic abnormality in a gene. Now, what have we been doing up till now? We're talking about, well, I did PGD. I found it has got, let's say, Tay-Sachs. I'll throw it away. I'll just discard it. I can't do anything with it. CRISPR says, okay, now we can look at this place where there's the genetic problem, and we can break, as it looks at the bottom, we can induce this double strand to break. So we open up the double strand, which is basically the double helix, which is the list of this information that we call DNA. We can take it out and we can fix it and we can put it back. So CRISPR and Cas9, what we can do is we can not just do gene testing, but we can do gene editing. We're able to edit the gene. We're able to now take out bits that we don't want and put bits back that we do want. And that's a completely different approach to genetic testing than we've done up to now. And the possibilities, the potential that CRISPR carries is just absolutely fascinating, because we can now take genetic abnormalities and we can fix them. Not only can we take genetic abnormalities that we think of, let's say, having disease like Tay-Sachs, but even things that we think are just not good enough. So we could do CRISPR and we could get, we could get, better bacteria, which will be able to be more effective, and we could get better fish, and we can maybe get better human cells. And we could get cells that will die less quickly, or they'll become anaploidic less quickly. And so we can actually use this treatment to change the world and to get a super fish and a super bacteria and a superhuman. And I was at a meeting a couple of years ago. Must be now. A couple of years ago meant when anybody said they says a couple of years ago, before COVID, because that was when we went to conferences. So before COVID, I don't remember what it was. I was at a conference, and a guy over lunch spoke about CRISPR. This was before Jenny Doudna had won the Nobel Prize, and he said that it was very, very new, and he said that excuse me, that there was a conference that was called in Southern California, that for the top biologists that were there, a few people that were hand chosen, that Jennifer Doudna had called. The person had said, did you come there? And she told the story at the conference of why she'd convened this conference, why CRISPR was important. She said she had a dream, and in the dream she'd gone to a world leader who'd asked her to come and explain to her CRISPR. How does it work? What can you do with it? And she explained to him when he's got his back to her. So he's explained to his back, to his neck, how CRISPR works and what he could do and what the potential. And he turned around in her dream, and it's Adolf Hitler, Yimach. And she says, "Oh my gosh, this is what we're doing. We can create the superhuman." And the guy said at the conference, the superhuman is one thing, but what we could create is even more frightening, is the super virus. And this was well before COVID. And now, we understand exactly what he was speaking about. Thinking that we could create a virus that was so dangerous, it could take over. We could take wheat that would destroy all the other wheat, or we could take a bird that would inject illness into other. There's all sorts of things we could do, and those are very much not really classified, not really controlled by any government. And CRISPR is becoming quickly the most quick developing and the most rapidly developing science in biology. And then every year you have hundreds of papers that are published on CRISPR, what we're able to do. And this is going to change our entire world. I don't know whether some of the older ones among us, I don't know if we're going to see that clinically available, but at some point, the younger ones here, and definitely the people who are in medical college, we give this talk to medical students, say, in your lifetime, you're going to see that's what's going to happen. That there's not going to be just looking at genetics. There's going to be, how can we write genetics? How can we edit it? How can we decide what traits we want in this embryo, what we want in this cell? Have an African minute for doing PGD in late onset. Okay, so just to conclude, so CRISPR at the moment is not really clinically available. It's really only things that have been doing in a lab. There was a scientist in Japan who started using cloning and CRISPR on human embryos, and in fact was caught and he was put to jail. He's in jail because it was, it's illegal to use it on humans. But it's only a matter of time. It's going to happen. If it doesn't happen in America and in England and Israel, where there's clear government controls, it happen in places where there's no government controls. What will be the ramification for PGD? We'll completely change PGD, because we're not going to discard the embryo. We're going to say, what do you want? What are the things that we want in this embryo? And we're able to put them in. And they don't, not only to get rid of, to eradicate disease, we could use it also to create a being that would be what we wanted. So it would be highly intelligent, it would live forever, it would be strong, etc., etc. So we don't sound, very doomsday, but we have to realize that this is the thing that's around the corner, and this is going to change every understanding we have of genetics. So if we wanted to just lesakem, what we spoke about, which is, breakneck speak about, we think, well, how far we've come from Gregor Mendel growing peas in his garden, and from Chazal's understanding of genetics, of how sisters are mechaskot, or have a chazaka of genetic abnormalities. We've come to the ability to be able to test people before they get married, to decide which marriages are wanted and which marriages are unwanted. We are able to give you indication of what you should be doing. We'll be able to speak about pregnancies, what will be the result of the end point of pregnancy, and we've gone even before that to what we'll be able to speak about before you get pregnant, look at embryos and to analyze them. And CRISPR will be able to do even more than that. It's to be able to actually change and edit and write genes to what we want. And so the question is, do we want perfect children? Do we want to get to that point that we're all perfect? Because there is a advantage of having certain genetic abnormalities, but who knows? That's a little bit of how do we can control and think about and analyze and maybe eradicate some genetic abnormalities. If anybody has a question, or a comment or a complaint, this is your time to do so. Otherwise, and we have a lot to think about in the next week about how we're going to use genes properly.
Amazing. I guess Yashikaya.
you. I'll send you I'll send obviously the PowerPoint, and everybody can listen to again in half speed, so we can. You get a 3-hour lecture, you can get in 3 hours. Shalom, shalom.